Can our brains continue to create new neurons as adults? Recent study sheds light on neurogenesis in the human hippocampus. Could it be true that adults have the remarkable ability to develop new brain cells? A groundbreaking study has provided compelling evidence that the human brain has the capacity for neurogenesis, or the growth of new neurons well into adulthood. This pivotal discovery promises to put to rest the long-standing debate about whether our brains can continue to adapt and heal throughout our lives. Let's science. This new neurogenesis glyph showed the lovely information you just needed to say that can grow new neurons. Furthermore, yes, the real power of ancient brains to bridge that can. But here's where it gets interesting. The creation of these new brain cells, specifically referred to as neurogenesis, occurs in the hippocampus, a vital brain region crucial for learning, memory, and emotional regulation. Explaining the significance, co-lead author of the study, Marta Padalini, a researcher at the Karolinska Institute in Stockholm, shared with Live Science, Kaidas en people Marta Padalini. In short, our work effectively resolves the long-standing question regarding whether adult human brains have the capability to produce new neurons. Indeed, other experts concur, affirming that the study presents a strong argument for the existence of adult neurogenesis. However, a single study cannot provide definitive proof. Yet, this offers strong support for the concept, explained Dr. Rajiv Ratan, CEO of the Burke Neurological Institute at Weill Cornell Medicine, who did not participate in the study. This is an excellent example of how great science is opening doors for clinical neuroscience, he said. A significant factor contributing to this momentous discovery is the usage of enhanced technologies. The researchers utilized advanced methods, including single-nucleus RNA sequencing and machine learning, to sort and analyze brain tissue samples from biobanks across the globe. RNA, which works with genes in a cell, and machine learning play crucial roles in processing extensive data sets to reveal hidden patterns and insights. Since the 1960s, it has been established that rodents and some non-human primates continue to develop brain cells throughout their life, particularly in the dentate gyrus region of the hippocampus. However, obtaining high-quality brain tissue samples from adult humans has remained a formidable challenge. Human tissue usually comes from autopsies or surgeries, and the way it's processed, how quickly it's preserved, the chemicals used, and the thickness of the slices can obscure these new cells, Padalini explained. By applying new technologies, our team skillfully overcame these obstacles. The researchers meticulously studied over 400,000 individual nuclei from hippocampal cells from 24 individuals, spanning ages from 0 to 78, including 6 children and 4 teenagers. Using 2 state-of-the-art imaging techniques, they were able to pinpoint where these new cells were located within the tissue. Astonishingly, they identified clusters of dividing precursor cells nestled among fully formed neurons, in areas where previous animal studies had identified adult stem cells. We didn't only notice these precursor cells in young children. We found them present in teenagers and adults as well, said Padalini. These cells possess the capacity to go through themselves and generate new brain cells. The emergence of these technologies enabled the research team to detect new brain cells at different developmental stages, a feat that was not possible only a few years ago. Dr. Ratan commented on the innovative approach. Using fluorescent tag, the team developed a machine learning algorithm that identified cells likely to mature into neurogenic stem cells, according to existing rodent research. In line with expectations, brains of children exhibited higher rates of new neuron growth compared to teenagers or adults. Encouragingly, out of 14 adult brains analyzed, 9 showed signs of neurogenesis using one technique, and all 10 showed new cells with another method. Yet, some brains have no clear signs of new cell growth. Padalini points out that the differences in adult brains still need more exploration to understand fully. Looking ahead, Dr. W. Taylor Kimberly, Chief of Neurocritical Care at Massachusetts General Brigham, who was not involved in the study, suggests further research into whether the neurogenesis observed in adults is connected to neurological conditions like Alzheimer's or correlates with overall brain health. Identifying these hidden cells is like finding needles in a haystack, he added, emphasizing their potential to enhance our understanding and track their presence throughout life. He envisions a comparison between dementia patients and superagers who maintain cognitive resilience in their later years. If a link between neurogenesis and disease is established, it could lead to innovative treatments. while specific therapeutic strategies in humans are still being explored, Patolini concluded, the knowledge that our adult brains can sprout new neurons revolutionizes our perspective on lifelong learning, injury recovery, and the untapped possibilities of neuroplasticity. So, what do you think about the potential for adult neurogenesis to enhance brain health and recovery? Share your thoughts and join the conversation below.